Hi there, I'm Dr Ben Britton of the University of British Columbia. This is the 7th lecture in the overview of Focused Ion Beam Microscopy. This lecture will outline some future directions I believe that the field will take with regards to FibSim analysis and tomography, sample prep, etc. And I'm also going to present a few of the capabilities that are currently, as of 2026, available or emerging to be available at the University of British Columbia. Of course, I want to acknowledge the contributions from a range of people, so I draw your attention to that list here. So let's look at some future directions. Telling the future is incredibly difficult, but we can look in the literature about where people are excited and opportunities and where they sit. So one of the areas of activity is to look at new source types. We've actually seen a few of these sources, or older sources, although they've been limited in the material systems that we're looking at. So we can have effectively things like the gallium source is a liquid metal or alloy-based source, and so that if you change and you have the appropriate material that has a low enough melting temperature, you can then use that to wet the surface of the tungsten filament and then enable that to have a liquid metal source. And that opens up a range of different systems. You can also have the plasma-based sources. We've seen that of those. That was how the xenon fib was formed, and there are a few alternatives that can be seen. The other one that we have seen effectively is the gas field ionization. That's how the helium ion microscope was being formed. That was having a very sharp needle, and then you use gas field ionization for atoms that effectively absorb to the surface of the tip, and then you get the trimer, and then you get the formation of the ion stream. I'm going to just show one relatively emerging source, which is the cold atom, the Motis source, in a moment, and there are a few others through ionic liquids that enable you to access. Ultimately, the benefit of using these different sources is we get to open up the periodic table to enable us to change effectively ion-matter interactions that are present, change things like implantation, evaporation, alloying, etc., etc. So the Motis source is the magneto-optical trap ion source. This process is super neat in that basically we can use a laser, and we can use laser cooling of the atoms to enable us to have effectively now that this ionization laser is joined with a series of laser beams to have a trap that enables us to then pull ions down the column and then use this as our ion source. And so here we can effectively now use lithium in our Motis. This source is super useful because you can be isotopically pure and you can increase the range of materials subject to you tuning the energy of the incoming laser in particular in this. And this enables us to do some cool imaging, so we can look effectively at lithium imaging of graphites, timbals and carbon in this for a 2 kV with a 1 picoamp, and we get these beautiful images that are present in this material. We've seen this a little bit as the emergence of FibSims, and here I'd say increasing application areas, specifically battery technologies, have motivated the generation and automation of FibSims methods. And so here we see one of the solutions. It's a test scan solution that's presented here that enables and is being promoted for the access specifically to look at battery materials in here. What we see here is that this is important for battery materials because lithium ions have no valence electrons, and so therefore it's a challenge to do other chemical analysis. And so instead you can collect this in the sputter yield, and so that's one of the areas that's being excitingly explored as the approach for lithium SIMS-based technologies. Of course, the challenges here are what was sputtered, how efficient was the yield, whether you can capture positive and negative ions, and how quick you can quant and automate. And so that's what effectively is driving some of these techniques forward in here. You can also imagine that there'll be integration with AFM techniques and other approaches, plus correlative approaches with things like nanosims and also, say, other macroscopic TOF sims and other sims approaches. Other areas that are emerging or have emerged, there's a beautiful set of work that was originally sort of developed at University of California, Santa Barbara, with Teresa Pollock's group, where effectively there's now a tri-beam system that introduces the electron beam, an ion beam, and a femtosecond laser. And the femtosecond laser lets us form ultra-short laser pulses that are highly focused and can eject the material. And then you can use this to effectively do large volume three-dimensional analysis, as well as attack certain materials that are less friendly with ion-based approaches. Here you can use the ion beam to clean up the surface or to do specific higher resolution work. You can link this to then use the use of EBSD analysis and other analytical techniques to enable three-dimensional analysis to be performed. And there's some beautiful literature. McLean, Eklund and colleagues have done a fair bit from Teresa Pollock's group, for instance, in titanium alloys, as nickel alloys as some examples. So this is then showing that you can have the femtosecond laser as an integrated approach that I sort of mentioned previously, and you can do this with, say, xenon or gallium sources, FIBs. But you can also set this up that you can have a shuttle to enable you to do femtosecond laser in a separate chamber and bring this under the FIB, so you can shuttle the sample back and forth. The balance of this is that effectively this is a tri-beam system. You have three things that can break, so you can have risk of downtime issues. In this system, you now have the femtosecond that can be used as a third-party tool for other cross-sectioning and other approaches. The other issue is that effectively this femtosecond, because it's reducing and removing so much material, this gets redeposited in the chamber and could cause you lots of redeposition issues that can cause some challenges. And so you might have to worry about protecting detectors or your chamber in these approaches. You can also use multiple ion sources in the FIB. This is part of the Hydra system that's found by Thermo Fisher, and so that can give you more versatile milling options. In some demo work that was kindly performed by Thermo Fisher Nanoport for us at UBC, there was effectively looking at the use of xenon and argon as the final milling steps for looking at an Inconel 718 sample. And here we can effectively see that the argon gives you a gentler final finish that improves, and here we can see the channeling contrast. That's what the mottling contrast is seen in the secondary electron image. The Hydra system can have lots of different ion columns, depending on which, and you basically shuttle the gases relatively fast to change those gases. And you can optimize the milling conditions for specific applications and materials depending on what you want to look at. Other future directions are the integration of broad ion beam technologies. I've shown this as one example previously with the Aura technology, but here we have a separate chamber, the iPrep attached, and this effectively is at the University of Manchester. And this was used to produce three-dimensional analysis where the broad ion beam system effectively is removing large areas of the surface, and there's a robotic transfer arm to shuttle it back and forth in the chamber. We can see the geometry set up, and that enables an approach to access large volume EBSD data for that particular analysis, as presented this by Ali Galinia and colleagues in this paper. We can also look at a range of areas where there are drivers, challenges, techniques, and approaches. There's a really good sort of analysis of this in Hofinger et al. In Applied Physics Reviews. And we can see here the drivers can be from different application areas, so life sciences, environmental, spintronic devices, quantum, etc. And we can flow through to which techniques are being used in the FibSem world. So subtractive processing, defect engineering, imaging, etc. And then we can see how the challenges are limiting the approaches in the driven areas. So for instance, we can go and say, let's follow through and look at quantum technologies, and we can see quantum technologies, they're often small scale pieces, so spatial resolution can be important, and spatial resolution can be addressed through things like novel ion sources, single isotope sources, and down to say better stages. And so we can see this linking of areas of where there can be innovations, and we see these as like highly modular systems, and we'll see different commercial competitors improving their approaches. Some of these things are going to be open APIs. We're certainly benefiting significantly at UBC with open APIs of this, and open hardware interfaces to enable us to integrate custom third-party solutions in here. Other bits and pieces, it can be just nice to work with the suppliers, and we're very fortunate to have a very good relationship with Tescan at UBC for my research group activities and our collaborators. So this opens us up into, let's look at the FibSem capabilities at the University of British Columbia. So we're quite fortunate that actually we have a number of different instruments across UBC. And so we have basically 3 test scan instruments, 2 of which I help look after, the AMBER X and the AMBER 2. There is another AMBER system that's in effectively the Quantum Matter Institute. And then there are 3 other instruments. So there's a Zeiss crossbeam instrument that's located in the bioimaging facility that's set up for gallium FibSem, tomography, imaging and analysis. And importantly, in this instrument, they effectively have cryo handling as well as an airlock system in there. In the Center for High Throughput Phenogenomics, Professor Nancy Ford and colleagues effectively have a Helios nanolab that's set up as a workhorse instrument, and this is set up to have a whole range of pieces, and we'll see the details of these instruments in a moment. The last instrument is located in the cryo-EM grouping in the Life Science Institute, and so this enables cryo-FIB and cryo-SEM for biological systems using the helium, it should be an Aquilos, apologies, it's an Aquilos FIB mill for this. So if we look at the capabilities, and I'm less familiar with the systems outside of my immediate domain, although I have used a nanolab a fair bit, but this system is set up with a gallium liquid metal source, liquid metal ion source, and it now has a quorum. You can see the handle here for sample loading. It has a range of analytical approaches: secondary backscatter, secondary electron, secondary ion, a STEM detector, EDS and EBSD. You can see a few of these detectors kicking off here. Has a platinum deposition system and is well set up for slice and view based imaging based upon the secondary backscatter Cesium images in here. And as I say, this is located with the Center for High Throughput Phenogenomics, and you can find the link here, and so you can look up and see how to access and costs and training effectively associated with that equipment. The Aquilos 2 FIB mill system, and I quote the instrument website. This is based in the Faculty of Medicine, and this is specifically set up for cryo-FIB preparation, for thin lamella, for subsequent T imaging in particular. So it's set up with a cryostage, SEM, and a gallium ion source, and it's integrated with an integrated fluorescence arm for effectively, you can do, I think it's luminescent electron microscopy, photoluminescence electron microscopy targeting, and can also be used for cryo-SEM of biological samples. And so that's a nice analytical setup for cryo and bio applications. And they've done a really nice job on their website to show a range of publications to give you a flavour of how those systems are useful in that. Within the Quantum Matter Electron Microscopy Centre, this is led by my colleague, Dr. Ismail El-Bahagi, and so you can find his stuff on his website that has more details. This is a test scan AMBER 1 instrument. It's a gallium FIB. Has a platinum gas injector, enables effectively slice and view through autoslicer, and it's really set up to enable lamella prep to support, in particular, to build out for the Nion instrument. There's a vacuum transfer system, and it has some analytics for EBSD, EDS, and backscatter electron imaging. And as I say, this is located in the Quantum Matter Institute. I think it's on the 3rd floor of the Brimacombe and QMI building for that. In the bioimaging facility, there is a Zeiss crossbeam instrument. This is a crossbeam 350. In this instrument, it's set up to have a gallium liquid metal source with a platinum gas injector. There's an EDS detector on it, as well as secondary and backscatter. And this is largely set up for tomography and lamella prep, and enables cryo-FIB and cryo-SEM through the vacuum transfer system and cryo stages in this. And that's found within bioimaging. They have a huge amount of expertise in analysing biological systems. I've seen some lovely work from their team. Now, moving more closely to the instruments that I look after in the electron microscopy laboratory, these are going to be physically based in the Brimacombe building. This is a new instrument that will be installed in Q3 and Q4 of 2026. We're waiting for the renovations to be completed. And in this instrument, this is set up largely to be sample preparation for TEM and micromechanical testing. So this is a gallium source. It's got a platinum gas injection system. It has an XYZR lift-out needle, a STEM detector, and the integrated argon broad ion beam system. And you've seen that beautiful example of the lamella prep as presented here. More details are located on our facility website. You've also seen in this presentation so far a significant amount of work from the Amber X instrument. This has now been operating for I think about 3 years, although we've just released it for full open access. There's a whole detail of how this works. And this is really set up in a fascinating way in that we set it up, fortunately, in an analytical plasma fib instrument, although it does do sample prep occasionally. It's built with a xenon plasma ion source. To support that, it has a tungsten gas injection system, an XYZ lift-out needle. We have a number of 3 backscatter detectors for some specific reasons, secondary electron and in-column secondary and backscatter, so lots of detectors. For the EBSD analysis, it's set up in the so-called static configuration that enables high throughput and high reliability for 3D analysis. We also have a large area EDS. We also do electron channeling contrast imaging. I'm not going to talk about that here, but we can do defect imaging in this. And we have the so-called rocking stage to enable us to reduce curtains in this. What's important also is that we've built out, and this is partially why this lecture series has been created, a full training pathway that's available to UBC folks via our Canvas portal. And this effectively starts off with people doing this series, the Introduction to Fib lectures. And then we go into effectively hands-on and lecture and quizzes to enable milling and deposition. And then depending on what you want to do, you can do a sort of a tomography pathway, you can do a lift-out pathway, and then you can do stuff to enable rocking. And then when we get the gallium fib online, we'll effectively also have stuff where we go into the auto-temp prep routes and the gentle ion beam-based system that will have the amber attached to this. And so this pathway effectively enables people to have piece by piece, and we're supporting a significant amount of offline and online learning, as well as hands-on, as well as getting people to really look at their own samples. And so there's been a huge amount of work, particularly by Dr. Ruth Birch and Dr. Simon Siankis on this. Of course, all of these facilities that I mentioned, or well, almost all the facilities that I mentioned, are located and supported by people who are working under the umbrella of the Characterization at UBC Research Cluster. And so I'm just going to close the lecture series by shouting out to this cluster and really encourage people to have a look at our facilities report and have a look at the range of activities and approaches, where the cluster is looking after in total around 46 or 47 million of financial investments across both campuses. And we have active collaborations from a fantastic team of principal investigators and also full-time staff underneath the facilities. We overlap with 9 facilities that are across the 2 campuses in here. The equipment spans X-ray, optical, and electron microscopy methods as our principal activities and the appropriate sample preparation routes that enable this to happen. And I create these lectures to try and help people really get the basics started so they can develop new approaches and really understand materials much better. With that, I just want to leave us with some overall take-homes for this course, that I've hopefully persuaded you that FIB is a flexible tool with many uses, and this opens up sample preparation, cross-section, tomography, analytical approaches, a whole range of other bits and pieces. Critical points to consider are things like ion sample interactions, optimizing the geometry of the experiment, and really about sample prep before it goes in the FIB. What I would suggest is that FIB is incredibly versatile, but it can be expensive, and especially if you make a lot of mistakes, and that's why getting some really good understanding of the approach, such as these lectures, hopefully will help you reduce the cost and reduce the stress associated with this. And I would also say that there are many alternative methods that can get you quite a lot of the way there that could be cheaper, but FIB is certainly a lot of fun to do stuff. There's a lot of resources, so I've referenced pretty much all of the figures that are here, but I'd also strongly recommend from the Australians the MyScope training series. And there's also a couple of other resources. There's this fantastic book that gives an overview of Introduction to Focused Ion Beams. You've seen a few of the pictures from this, and there's this other website that provides some more details. And I'm going to plug, of course, that we have a fair amount of bits and pieces on the EM Lab and our training courses for people who are going to use the Amber instruments that we've got. With that, I want to thank you very much for your attention, and of course I welcome questions or other such as time progresses.